So we are going to start today talking about the role of the pancreas and the liver in the regulation of digestion. So to start with the pancreas. So the pancreas is both an endocrine and an exocrine organ. So you can see the two types of cells in this cross-sectional image here. The yellow cells are the islet cells, which act as that endocrine part, and the hormone secreted, specifically insulin, which we will talk about later, is released into the bloodstream, and you can see the capillary in the image. The exocrine role comes from the acini, which look like the lobules here. The secretion of sodium bicarbonate comes not from the acini, but rather from the duct cells, which are along this area here. Digestive enzymes are released from these lobules into this duct, which leads directly into the duodenum. Sodium bicarbonate travels this route as well. So insulin is one thing that was mentioned that the pancreas releases. Again, that's through the islet cells. Insulin is synthesized by the pancreas, but it needs to be activated before the secretion. The stimulus for the secretion is glucose, which is found in the plasma. When glucose is taken up from the small intestine, it is taken to the pancreas by a GLUT2 transporter, which causes a depolarization and exocytosis of insulin. Another thing that happens in the pancreas is digestive enzyme secretion. Again, these are secreted by the acini lobes of the pancreas. These also are released as zygomins, which means they must be activated once they reach the intestines. So their release is activated just because there is food in the small intestine. They could also receive neural signals or be released due to the presence of CCK. So this picture here talks about sodium bicarbonate. Sodium bicarbonate is important in the digestive system because it neutralizes the acidic contents coming from the stomach. Its formation is slightly complicated, so that's why I added this picture here. So the mechanism of action is shown, but I did want to point out one transporter, and that is the CFTR channel. So that channel is right here. See, it listed right here, that's CFTR. And what this does is it allows the Cl to leave this cell, which later re-enters in exchange for that HCO3. And this is the pathway that leads to the disease cystic fibrosis, which is characterized by thickened mucus, which is blocking the pancreatic duct, so digestive enzymes cannot be secreted. Next, we will be talking about the liver, and the liver itself is broken into two main lobes that break down further to connect to ducts, which come together to form the common hepatic duct. This will come into play later when we talk about the role that liver plays, but the secretory part of the liver is the hepatocytes, or the liver cells. They are hexagonal in shape and secrete bile salts for fat digestion, better known as bile. So as mentioned, bile salts are made in the liver, specifically the hepatocytes. Now, the bile salts are made from something called bile acid and amino acids. So after secretion, they travel into the gallbladder for storage and concentration through the hepatic ducts. So here's the gallbladder here. You can see in this picture it is nested right underneath the liver. And then it's from the hepatic ducts that, you can see it tucked back here, that bile enters the gallbladder. Now it is said that you could live without the gallbladder, and the bile salts, or the bile would just be directly secreted into the small intestine then. Anyway, so they are signaled, the bile is signaled by fat in the meal, and the gallbladder contracts to release the bile into the duodenum. Bile salts break down the large clumps of fat that enter the small intestine. They come in as large clumps because they're not water soluble. So in the stomach, instead of being broken up, they actually come together to form a large clump. The bile salt then breaks down these fats, and the newly broken down fats are surrounded by bile salts, creating a micelle. The micelle then goes on to further break down by lipases, and there's a colipase that is used to remove the bile salt from around the micelle so the lipase can get in and go break down the fat further. So after digestion of the fat, the bile salts can be reused, and in order to be transported back into the liver, they are broken back down into bile acid. This is important because in a singular meal, you don't have enough bile to break down the fat, so you need to reuse those bile salts multiple times over in a single meal. Lastly is the filtering role that liver plays in digestion, and the liver is sort of a dump site for blood coming from the digestive tract. So you can see it coming here, and it goes into the hepatic portal system. So hepatocytes, which again created that bile and bile salts, so besides that secretion, they also release enzymes for metabolism of drugs and other toxins. So this is to make sure that these toxins are removed from the blood before going back to the heart for circulation. Thanks for sitting with me today. Here are a few references, including the pictures if you wanted to go read more information on them. And then all of the information on my talk itself came from the book, The Silver Thorn, which is right here.